Good afternoon, everyone, and welcome to our last WeedSmart webinar for 2019. I'm Shannon Barrett, I'm Communications Officer here at WeedSmart. To begin with, I'm just going to introduce WeedSmart and do a quick bit of housekeeping, and then I'll hand over to Paul McIntosh to introduce our talent today. WeedSmart is an industry-led initiative delivering science-backed weed control solutions to Aussie growers to keep us cropping. We are industry funded by the major chemical companies and have GRDC as a major investor. As everyone can see on the screen now, everybody above the orange line is a financial investor and everyone below the line is an in-kind investor. I'm now going to hand over to Paul McIntosh, who's our Northern Extension agronomist, to introduce Bhagirath.
Thank you very much, Shannon, for that, and thank you, Lisa, and good afternoon, everybody in Australia who's listening as overseas. I'm sitting in the Gunnedah Bowls Club, and it's in the non-drinking section. I'm in a small office and it's no air conditioning, and I think it got to about 39 degrees here yesterday, so I'm sweltering, so in keeping with the rest of the country. But on the line for today's webinar, we have Bhagirath Chauhan. I call Bhagirath Sunny because it's a lot easier, he said. Sunny is the Principal Research Fellow and Team Leader at University of Queensland, based down at Gatton there. He is part of the QAAFI syndicate over here, and he's got so many things to his bow. He's got Associate Editor of Weed Science, Editorial Board, Associate Editor Indian Journal, and of course Special Editor of the Weed Management Frontiers in Agronomy. So he's a well-cited researcher, and we're lucky to have him this afternoon. And of course we're going to talk about those two real pesky weeds, to coin a phrase, called Feathertop Rhodes and Barnyard grass. And some of the ecology or biology that Sunny's coming up with will allow us to handle these particular weeds better in the future without relying totally on our herbicide suite. So I'll pass over to Sunny and we can get some good information on those two pesky weeds that trouble us so much in our summers.
Just before you start, Bhagirath, can I just get you to click up on the display settings and change the screen so it's the full? Perfect, thank you.
All right, thank you very much, Paul. Good afternoon, all of you. Now, I started my presentation with this slide, and there is one quotation. It was in 1968, that weed-free environment is almost with us, with the use of herbicides only, and there is no reason why it should not be possible in the next 10 to 20 years. But what happened after 50 years? Have you got rid of any weed? No, I don't think so. So what happened then? We have got now more weed problem. So what we need to do, I think just reliance on herbicide is not enough. That's why my presentation today is about weed biology insights to improve management of these two weed species, Feathertop and Barnyard grass. Before I start this presentation, I would like to thank GRDC for funding this research. So I'm presenting results from our two GRDC projects, plus some of that previous published paper. Little bit background, I don't want to go in details what are these weeds, but just to show that these two, barnyard grass, if you see the left-hand side graph, it's mostly in the eastern side or right-hand side, and Feathertop Rhodes grass, it's more or less uniformly distributed throughout Australia. As I mentioned earlier, herbicides are now our dominant method of weed control, and therefore heavy reliance on herbicides. So what happened then? Evolution of resistant weeds. And this herbicide use, it has coincided with concerns over hard to control weeds, weed population shifts, increased cost of chemical control, and environmental pollution. So that means we need to develop ecologically based integrated weed management programs. However, to develop such programs, we need better understanding of weed biology. And that information will guide us to enable successful long-term control management. I just want to point out one important thing. Some people who are working on herbicides, they think that, okay, just only herbicides can provide you good weed control. But I think the research on weed biology, that does not provide you any product. It's mainly results in procedures or knowledge rather than products. So I think we need to understand that one before I go in this detail. Okay, let's start with Feathertop Rhodes grass. There are 3-4 components I will talk later on, but first just see effect of weed density on grain yield of mung bean. In this graph, if you see on y-axis, it's grain yield of mung bean in kg per hectare, and on x-axis is weed density of Feathertop Rhodes grass density. We did this trial in 2 years, and if you see this on x-axis, Feathertop density, that was from 0, which is weed-free condition, low, medium, and high. And from this trial, we found that only 22 plants of Feathertops per square meter were needed to reduce 50% yield. And in this study, we used actually 50 centimeter rows. But if you see growers, they follow up to 1 meter. So what that means, we may expect higher yield losses due to Feathertop. So this is just to show you the importance of this weed. Now, a little bit about germination response to temperature. And I think there is a particular reason why I want to discuss about this particular topic here. If you see this graph, on y-axis, it's germination, just percent of maximum. And these all different lines are different populations. This is not really the right method of presenting graphs, but I want you to follow that particular one line so that you can see that germination at different temperatures. And on x-axis, it's temperature, day and night. Like for example, 15.5, it means 15 degree during daytime and 5 degree during nighttime, so 12 hours. Now you can see Feathertop Rhodes grass, all populations, they germinated at all different temperatures, ranging from 15.5, which could be our winter, mild winter temperature, and to 35.25, 35 during daytime, 25 during nighttime. So seeds germinated, what does that mean? That this Feathertop Rhodes grass now can germinate even in winter. Again, we used 4 or 5 months old seeds here. That's why I mentioned after ripened. So people may say that, oh, they might have lost their dormancy. So that was interesting. So what happened then? This year in April, we collected seeds from different parts of Queensland and New South Wales. And then immediately after that, we planted in pots, but there it was irrigated condition. And these are only 8 population. You can again see y-axis germination, and on x-axis is time immediately after planting, and they were planted in outdoors. And you can see that seed germinated even in those winter months. And if you see, germination ranged from 13 to 83, depending on population. Again, if you see this graph and this gray line, that's temperature. And where germination occurred, you can see that temperature was sometimes 0 also. So 2 things. Seeds germinated during winter months also, and 50% of seeds germinated within 2 weeks. What does that mean? I think until now, our research showed that this weed has got 2-month dormancy. But that may not be true now, or even, but it depends on population. You can see some population had more than 80% germination. So this means that Feathertop can germinate and emerge under field conditions if there is any rainfall events in winter also. And these results are supported by current winter season observations by growers and consultants when they saw this weed in their wheat crop or just in winter. But we are not sure if they will grow successful and produce seeds, because I think frost may kill that. But we started a trial here where we are planting this one every month. Also, what's the implication of these results? One is, yes, if they germinate, their seed bank may deplete if there is good moisture in winter, and then therefore there will be less density of this weed in summer crops. But on the other hand, it also indicates possible adaptation. I don't know whether in the past we observed these responses, but this weed again may become a problematic weed throughout the year, like saw thistle. It was mainly a winter weed. Now we can see that one throughout the year. And also we need further research to fully understand why this difference in germination, because between different populations. Is there anything, Paul, you want to discuss?
Well, Sonny, I'm just, I've seen the notes you said. We discussed this before we did the webinar today, but that's very interesting that we're lining it up like a problem like South Thistle, where it seems to be growing nearly 12 months of the year, or it seems to be, is growing 12 months of the year. And of course, the distribution of with Feathertop Rhodes all over Australia really does bear out a really major problem for Australia nearly 12 months of the year. So it's good research work. It's good to know that, and good to know that there's no dormancy too. I think that was the other point you brought out there.
Yeah, I think that also depends on population, because there are a few populations. We don't know what's the, these populations were collected from a particular cropping system, whether there was any maternal environment which affected dormancy. So I think these studies are in progress.
That's good.
Thanks.
Those cohorts from different locations, so that's a big thing. You know, we all grow our own problems, but they may not be the same problems as someone, 100 miles down the road or up the road.
Yep, totally agree. Yeah. All right. Second component is seed production and seed retention. I think this particular component is about harvest weed seed control. Nowadays, we hear. So if you see this graph, on y-axis, it's seed production, and retention is given in percent. And this study was conducted in mung bean. Feathertop Rhodes grass was grown in mung bean in 2 years, 2017 and 18. And what happened then at harvest, we found that this weed produced 120 to 148,000 seeds per square meter. But seed retention, it means seeds still intact on the plant of Feathertop Rhodes grass, you're 93 to 97%. It means only less than 7% seeds were dispersed at crop harvest, and which points to the possibility to manage Feathertop through seed catching and destruction coinciding crop harvest. That's in mung bean. But when we go in sorghum, this is sorghum graph. Again, first I want to explain here, y-axis again, it's seed production, but this is per plant now. Okay, and seed retention is given in percent values. On x-axis, we planted sorghum one time, but then Feathertop Rhodes grass emerged in that crop at different times. With the crop, which is 0, 2 weeks after crop, which is shown by 2, 4 means 4 weeks after crop emergence, 6 means 6 weeks after crop emergence. And we evaluated seed production and retention. Okay, I want to mention here that our growers only harvest like panicles of sorghum. But in this case, we harvested this sorghum at 15 centimeter above the soil surface, just because we just wanted to know whether this weed has got potential for harvest weed seed control. And what we found that these first 2 cohorts, when this weed emerged at 0 and 2 weeks after planting, they produced around 5 to 6,000 seeds per plant. Okay. But for this cohorts, which is 4 weeks after sorghum emergence, that produced only 355 seeds per plant, and no seeds for the last one, 6th one. Okay, and what that means, it suggests importance of early weed control. If we could manage these weeds for first 2 weeks, for example, using any pre-emergence herbicides, then I think this this weed is easy to manage. And these late cohorts, they were affected more by competition from these larger crop plants. If we talk about seed retention, which is in percent value, that I think 67 to 75% for these first two cohorts. And it's again suggests there is a potential for harvest weed seed control tactics. Also, these results suggest that if we delay crop harvesting, that's not really a friendly practice, because I think seeds are likely to be blown by wind. Okay, and
that's a good slide, that one, Sonny, with the use of metolachlor in our grain sorghum crops. You know, we know that it gives about 6 to 8 weeks control of grasslands when under moisture, and it's a critical period for getting control of weeds, as you've highlighted it with Feathertop Rhodes in a grain sorghum crop.
Yeah, again, I want to mention one thing here. I think it's irrigated condition, and we planted at 1 meter row this particular crop, and there were 7 plants per square meter, around 70,000. So I think that also we need to see about a competitive crop, but I will discuss more about that one later on. Okay. Now, next is Phenology. I think it's very common amongst crop scientists, but I think for weeds or for us, just in simple term, what is this one? It is just study of a plant life cycle or their events. For example, days to flowering or seed production, and how this plant life cycle is affected by environmental condition. So mainly, for example, just one weed, barnyard grass, that germinates from September to March. But Phenology is when it germinate or emerge in September versus in March, when they are producing more seeds and what's their flowering time. So this is just simple study there. And it's mainly affected by temperature, photoperiod, and moisture. But for this particular study, I think we will take out moisture because our studies were conducted in irrigated conditions. We did some in rain-fed, but sometimes there was no rain, no moisture, so plants died. So I will present mainly related to temperature. Okay, before I go into that detail, just there are three. First of all, in sorghum and feathertop, you need to understand here, these both species were planted 4 times: November, December, January, and February, planted together. But feathertop was planted, just if you see the far left, without a crop. So we simulated like a fallow condition, okay, when there is no crop there, single plant. Middle one with 1 meter row crop, and then with 0.5 meter wide crop. Okay, they were planted at 4 times. Now, if we come to the results, y-axis, if you see in this graph, that seed number per plant. I think, and there is one, you can see in that y-axis, there is one break, and that one, just so that you can easily understand, that's 40,000 and it goes up to 210,000, okay? And on x-axis, it's planting time, 10th of November, December, January, and February. Okay, and there are 3 different graphs. I will talk that one later on, but let's see first, potential seed production. I think until now, our report said that this plant produced only 40,000 seeds, and that was in South Australia, per plant. But in our condition, we found more than 200,000 seeds per plant. And I think this one, for example, we had around their panicles or inflorescence around 150, and each one had more than 1,000 seed. Again, I want to mention one thing. This is irrigated condition, so ideal condition. Second, we actually counted all seeds, whether they were mature or immature. So there were around 25% mature seeds. So at that time, so it means out of 200,000, around 50,000 seeds per plant. So you can see that potential of this particular weed. So this can produce lots of seeds. So it means isolated plants in fallows must be controlled before maturity if you want to prevent seed set. And that could be through any different tactics. And most effective management is to prevent seed production, drive down the existing seed bank through different management practices. Okay. So this study, again, it was conducted in 2 years, and I'm showing just 1 year data so that you can easily understand. Second component of this one was Phenology. So when we planted in November, December, January, February, I think that's November planted cohorts produce the maximum number of seeds, irrespective of crop competition. You can see these all 3 different graphs. Here, I think it's not really about, you need to remember month. It's mainly related to temperature, nighttime temperature. So that may differ, like in any year, it could be like December, but it won't go in like February, okay? So this is affected by temperature and photoperiod. So it means we need to give more emphasis on early cohorts because they produce the maximum amount of seeds. Third component about this study was crop competition. Okay, again, if you see this left-hand side, if you see the left-hand side figure, there are 3 photos here. I just, it's not really that ideal, but front one, if you see, this Feathertop Rhodes grass plant, when it was grown without any competition. So you can say in fallow, for example, you can see its biomass, seed number. Middle one, it's 1 meter wide crop. And the last one, for the, it's a 0.5 meter wide. So you can easily see the size of Feathertop Rhodes grass. Okay, and even if you see the graphs, in that one, November, December, if you see these all dark colored graphs, One is 1 meter wide crop and another was grown in 0.5. See, almost more than half, 50% reduction in seed number. So these results suggest that narrow row spacing expressed Feathertop Rhodes grass seed production more than wide row. And I think there is no surprise. We know this one crop competition we can manage. But what I want to suggest here, if opportunity arises, use narrow row crops. Again, I want to show that figure again so that you can easily see crop with Feathertop Rhodes grass without crop, with 1 meter wide crop and 0.5 meter wide crop. Paul, anything is that related to that?
Comment there. It's a great slide, a great bit of research, Sonny, about the use of crop competition. Of course, Peter Newman, one of my AHRI and WeedSmart colleagues from over at Geraldton, this has been his #1 catch cry for many years. He loves his crop competition, providing better weed control and better weed seed or lesser weed seed production. So it's spot on to what Peter's been advocating for many years in the winter crop growing areas. And of course, now that this research is out with yours, it highlights that we can do the same thing over here with crop competition with some of our weeds, as you've shown there on the slide. So that is good.
Yep. I just related to that. I want to show just only this slide, Feathertop height, when they were planted in November, December, January, and February. If you see the November one, this Feathertop Rhodes grass was almost 2 meters. Okay. And if you see this tree, when we increased crop competition, its height actually increased. So early cohort stall provides that if their height is increased, that means enhanced crop competition will increase their height. It means that's better for harvest weed seed control. Okay, that just I want to emphasize here. In sorghum also, we can harvest that one, this Feathertop Rhodes grass. Okay, next one is seed bank persistence. Now, after phenology, we talk about a little bit about their seed bank. But before that, I just want to mention that what's their effect of burial depth on emergence. I think maximum germination for most of these weed seeds we find from surface seeds, but there should be enough moisture. Okay. In our study, we never found emergence from 5 centimeter depth, but in a South Australian study, they found that around 10% seeds could emerge from 5 centimeters. These results could differ. I think that's not really point of discussion here. 10 or 5% is not really the problem because it all depends on researchers also. What's your bulk density of your soil type? But what I want to suggest here that a strategic tillage operation that buries seeds below their maximum depth of emergence could be a management option, mostly in case where seeds have concentrated on the soil surface. But if you see this another slide, I think it's all about its seed bank life. So there are 2 graphs here, 2 populations we used, Dalby and St. George. Both had similar results, so forget about this left-hand side, just concentrate on the right-hand side. Also don't need to see these all different lines, just see these 2 circles. The first one is a green one. So seeds at this is surface, when seeds were placed at the surface or at 0 centimeter. So these seeds depleted at a rapid rate. So most of them, all seeds actually, they depleted within 12 months after placement. But for buried seeds, like when they were buried at 2 or 10 centimeter, around up to 15 centimeter, 15% seeds were still viable at those depths. But regardless of burial depth, I think all seeds depleted within 18 months, 1.5 years. Okay, so that's a good thing that this weed species cannot really persist for that long. But on another thing, I think in South Australia, what they found that buried seeds can persist for longer if they are dry. So like if we experience in this year, dry years, they may persist longer. Also, don't really think that seed burial could happen only during cultivation. It also happened during planting operation. Also, this time in some soils, we have got big cracks. So seed could be there in those cracks also. Okay. Also, what these results suggest that this population, as I mentioned, these feathertop are unlikely to develop persistent seed bank in no-till, and that could be reduced quickly if we don't allow other seeds to be added in the seed bank. Okay. But as I mentioned earlier, it is this plant can produce a large amount of seed. So even a small seed bank can lead to successful colonization of agricultural as well as non-agricultural areas. Just related to that, this was initially not in my presentation, but I think now we hear a lot about this high temperature going on. What do we know about seed bank persistence? Again, we don't really have direct data on that type of climate or weather condition because we never had in the past. But I think here I want to mention a little bit one study we did in a lab condition. Two things, seeds we used were dry, so without any moisture. And in another case, we pre-soaked seeds for 24 hours. What does that mean? That they absorb water. Okay, just see these two things, dry seed versus wet seed or pre-soaked seeds. And we place these seeds at high temperature, like 40 degrees, 70, 100, and 130 for 5 minutes and check their germination. Okay, we don't need to go in detail what was the temperature, but what it suggests that to reduce 50% germination, around 90 degree was needed for pre-soaked or wet seeds, but for dry seed, it was 120 degree. Means that we need high temperature to desiccate these dry seeds. But this particular study we did just to see if the narrow window burning could help or could kill those seeds. Also, I think effect of this current dry and hot weather on seed viability is not clear, may have little effect, but in my view, it's not enough to manage. Seeds should be imbibed if they to die. And there are some of my views, these are my personal opinions related to this type of research. To me, I think increased temperature and moisture, they are the most important to seed germination and viability. The more moisture, the more kill, or you can say germinate, and then we can kill them. Okay, so there should be moisture. Seeds in dry soils are usually tolerant. If it is totally dry, I think up to 60 to 65 degrees centigrade. For particular, this feathertop I'm talking about. Okay, and I think Paul already observed around 62 degree a few years back on the soil surface. So that particular temperature, I think, may not be able to kill. But seeds in moist soil, they lose viability very fast. It could be through germination or decay. So what I mean, seeds should be imbibed. Another thing which I want to mention here, it's not related to feathertop or barnyard grass, but I think you will expect that hard-seeded weed species will lose dormancy faster. So at this high temperature. So expect higher germination of these species when we have enough moisture. And some of these species are marshmallow, cider, wild jute, bladdercatemia, susbania, cow pitch vine. How I selected? Because these weeds, they have got hard seed coat. Okay, so if you don't scarify their seed coat, germination is less than 10. So their seed coat need to be scarified. And I think that's why for these particular weeds, this high temperature will soften their seed coat, and when they get enough moisture, they will germinate. Again, feathertop would survive on the soil surface. I think that's in my view for at least 2 years. And before I finish this Feathertop Rhodes grass and I go to Paul, I just want to show this particular diagram so that people can understand how the seed bank dynamics work. I don't know whether you can see my cursor, but this is a seed rain. These plants, they seed dispersal through different methods like rain or wind, animals or machinery. And then we have got a pool here of seed bank pool, you can say. It has got all different kinds of seeds plus dormant seed. Some of them, they decay through fungi or pathogens. Some, they are taken by birds, mice, or insects. That's all good. Then third component is germinate and die. And this is where I think if you have, for example, one good rain, and then you have got dry period, that happens there. The seeds will imbibe, they germinate, but then they don't have enough moisture, and then they will die. And these all three things I think we should really encourage. But then the last component is germinate and establish. There, I think we always go after, for a herbicide, or we try to kill using different methods. So this is just seed bank dynamics. Okay, Paul, is there anything related to temperature or seed bank persistence?
No, but I can confirm that temperature of yours on the ground. People mightn't have done that, and I put a soil thermometer on the ground north of John Darien at Barnyard back in 2015 in January, and it was burning me through the knees of my trousers, the long trousers, and it was 62, 63 degrees centigrade on the soil surface. So it's a bit scary to figure it can burn my knees and make it a bit uncomfortable to lie there, and not has any positive effect on those seeds unless they got some moisture inside them, Sonny. So. Yeah, I think we all hope that high temperatures on our soil, particularly our dry soils, would be decimating some seed populations, but it doesn't appear to be doing that for Feathertop Rhodes, anyhow.
Yeah, I think third year we are not sure. Definitely it could, and we don't want that we have this dry weather the third year. We need this rain. But I think Feathertop, we also need to see, it's very complex. We also need to see the size of the weed seed, also moisture. If these weed seeds have got high moisture, then they will desiccate faster. If they have low moisture, I think there's less chance. So it's very complicated. And I think even if somebody say that 50% will die, and I see that seed number, even 90% die, that 10% is a lot. So I think we may have less population, but definitely seeds will be there.
Very true.
Yeah. Okay, next one is Barnyard. I think it's more or less similar results, but some are different. For example, we discussed for Feathertop Rhodes grass about temperature. That can germinate even at that lowest one, 15.5. Here, I think just see on y-axis for Barnyard grass, we used two populations actually. And this is all about these two populations. One was glyphosate resistant, another was glyphosate susceptible. Again, they had the same genetic background because we actually developed this population using clone method. Okay, we don't need to go in that detail, but what I can say here that in y-axis it's germination, x-axis it's alternating temperature. So 5-15 minutes, 5 during night time and 15 during daytime. So what happened in this one, seeds did not germinate at this temperature, 15.5. So I think that's one we can see in the field also. When we have winter season, we don't really see germination of that. Now there are two graphs here, two bars here, light and dark. Light is where germination in light condition, and dark means germination in dark. So what does that mean in the field? Light means when most of seeds are on the surface, so when they receive light, sunlight. Dark means when they are either buried in the soil or they are under a crop residue cover. Okay, now you can, I think, relate these results with that one. So germinated, this particular weed germinated from 10-20 to 25-35. So I think there is considerable opportunity to emerge over spring, summer, and autumn. And I think this is not surprise. We have seen that one. Light increased actually germination, and that's also we see in the field that in no-till condition there are there's more germination compared to cultivated ones. Another interesting thing what we found here, if you see this dark bar, germination actually increased with increase in temperature. You can see that one increasing from 10-20 to 25-15 and 20-30. What does that mean? That indicates that these seedlings may have a tendency to emerge from buried seeds in the summer season. So it means initially you may get the most germination from your surface seeds, and later on when your soil warm up, then I think we may see this germination from deeper depths also. And again, these results also suggest that we use lots of this conservation agriculture or no-till farming. So even crop residue retention may not really going to suppress this particular weed species. Again, seed bank persistence. We do not have our own data on this particular weed, but I think 6 months ago we started using 3 populations. They are glyphosate resistant and glyphosate susceptible, and at 3 locations: Narrabri, St. George, and Gatton. So they represent different soil type and different rainfall. So I think in the next 1 or 2 years we will have data from our own trials. But here I'm just presenting from Steve Walker's like 10 years back when they did that one. Similar story, I think short persistence in the surface layer, but it's not that short. I think here for barnyard grass that persisted for about 2 years on the surface, and buried seeds, that was around, I think you need to see only these 2 lines with this green oval or green circle. So this top one, there are around 24 seeds persisted when they were buried. Okay, so what these results mean that it's preferable to leave weed seeds in the surface layer, or do not, if you need to till, just wait, don't till immediately after harvest, as this will lead to a more rapid decline in seed bank. So if a paddock with the resistant population, so any management strategy that prevent weed seed burial and seed drain will lead to a rapid decline in these populations in the next over 2 to 3 years. Okay, seed burial, I think this study we do usually in controlled conditions. We provide the ideal best condition, irrigated condition. We just want to see what's the maximum depth of emergence, okay, where they could emerge. So I think we did this study 2 to 3 times, and we found that this particular weed, okay, this graph is y-axis is emergence, x-axis is burial depth, and that could emerge up to 6 centimeter. Okay, it never emerged from 8 centimeter. And again, we used 2 populations, glyphosate resistant and susceptible, and there was no difference between these 2 populations. So again, it's the same story like Feathertop, strategic tillage operation that buries below, these seeds below their maximum depth of emergence could be a management option. And especially I'm talking about where seeds have concentrated on the surface. Okay, for Feathertop, we had data of seed retention in sorghum as well as in mung bean. But for this one, barnyard grass, we don't have at the moment in mung bean, but I'm presenting only in sorghum. Again, if you see y-axis, it's seed production per plant, and retention is given in percent value. Okay, and on x-axis, okay, we planted sorghum 1 time, but then barnyard grass emerged in that crop at different times after crop. So, for example, 0, 2, 4, and 6 weeks after crop. If you see the first cohorts which emerged with the crop, that produced around 4,000 seeds per plant. But if you see just only after 2 weeks, what happened then? Almost 75% reduction in seed production. And then at 4 weeks, only 80 seeds per plant, and then only 30 seeds per plant. And this is again the same story. It's not like Feathertop. To me, from this data, it looks like Feathertop was more competitive than barnyard grass. But this one also again suggests importance of early control using pre-emergence herbicide, as Paul mentioned, use of Urochloa or dual gold. Second component is about seed retention. If you see the percent value, seed retention here was 42 to 46%. It's not really that great. Like for Feathertop, we had around 75%. And these were only for early cohorts. So this suggests that we have limited opportunity for use of harvest weed seed control in sorghum. And this one is mainly, I think that's my personal view, most of our summer weeds, they mature and produce seeds within like 40, 50 days. So I think that's time difference. In sorghum, to me, it may not be possible. Because I think this barnyard grass that produced seeds and shattered already within 50 days, but sorghum was harvested around after 100 days. Same story maybe for other species like windmill grass. So I think we need to see we need to see a possibility in mung bean actually. Okay, so research required to evaluate seed retention if you want to go in that direction in other summer crops which are actually short duration like mung bean. Okay, this one again is a sorghum competition, as I mentioned earlier, similar to Feathertop Rhodes grass. And here again, I think if you see this picture on the left side, when barnyard grass was without any crop, single plant, then in 1 meter row crop, and the last one is 0.5 meter. Okay, again here, sorghum and barnyard grass, both planted in November, December, January, and February. The results are very similar, but I think it's if you see here, I think on y-axis it's seed number. Again, there is a break in that axis. After 45,000, it's 60,000, just to accommodate these small values, which are like 95. So what these results is that with no competition, if you see December or January cohorts, they produce maximum amount of seeds, 75,000 or 45,000. Again, I think that suggests importance of weed control and fallow conditions. So we need to give more emphasis on December, January. And I think, as I mentioned earlier, don't just go at this after this month. This is all related to temperature. So we have got the, if hot weather, I think we need to, these weeds, if they get good moisture, they will produce lots of seeds. So we need to, and yeah, if you see this, again, second component was about crop competition. If you see this photo on the left-hand side, first plant is about without competition. This is barnyard grass. You can see its biomass compared to the second one, which was in a 1 meter wide sorghum crop, and then third one is 0.5 meter wide crop. Again, if you see this graph, and these two dark, blue ones, if you see 1 meter and 0.5 meter crop, it's 50% reduction in seed number. Again, as we saw in similar to Feathertop Rhodes grass, here also narrow rows expressed barnyard grass seed production more than wide rows. I want to go again with that figure again so that you can see that those plants, when they grow without any crop and with 1 meter and 0.5 meter. Again, I want to emphasize these were in irrigated condition, so ideal condition. But I think crop competition will do the same thing. This one last component, but one thing I added again yesterday when I discussed with Paul, and they suggested that yes, if there is any fitness penalty, we need to discuss. These trials we did, I think first of all, I want to mention that this was in the pot, and barnyard grass was planted without any crop and with mung bean 4 or 8. Don't go after what that means, like 4 and 8. It's just only like when they grow without competition and with competition. We wanted to see, is there any fitness penalty? Second is, I think people, they talk about fitness penalty when they compare one glyphosate resistant, for example, from Dalby, glyphosate susceptible from St. George. That's not really fitness penalty. I think you need to compare from the same genetic background. And that's what we did here using the clone method. It was from the singles population. And what we found that growth and seed production were similar for both biotypes, and which suggests that there was no fitness penalty. Again, I think I want to emphasize again, this was only one population. We may have different results if we use, for example, 10 populations. So I just wanted to mention here that for this particular biotype, we did not have any fitness penalty, okay? And this is just showing averaged over biotypes. Top graph is seed number, and bottom one is total shoot biomass. This is gram per plant, just showing importance of crop competition. When you have, when plant is growing without any competition, they produced around 30,000 seeds per plant compared to, with 4 mung bean plant or with 8, and that reduced, seed production by 85 to 95%. Again, this is an ideal condition, but in the field also, that shows the importance of crop competition. Again, I think when we say crop competition, don't really think that this is only the one method or it could provide you 90% control. No, I think for me, my personal views, if any of these one tool could provide 10, 15, 20% management of control, I think I would be happy, because I think our emphasis should be how to best use these herbicides or how to integrate other tools with herbicides. Okay, so that's why I think we need to integrate with other methods. This is last slide before my summary slide, but this one is not really related to weed biology, but I thought I should show you because I think we did it recently. It's a moisture by glyphosate efficacy study. And we used 470 glyphosate, and on the left column, it's first column, it's glyphosate in milliliter. And I just want you to see well-watered and water stressed. There are two main columns. Well-watered means when these plants were watered every day, daily. But in water stressed, irrigation was stopped 2 weeks before spray. I'm sorry, I don't have really exact data like how much was that water that time, because I think it was done by my student. But I think what I want to show you here, that we use susceptible population. If you see in well-watered condition, susceptible at 1.5 liter, there was no survival. Plant did not survive at all. And also at 3 liter. But if you go in this well-watered, yellow highlighted area of water-stressed condition, survival was 90% and then 25% at 3 liter. Same with when the registered population, it was only 19% at 3 liter, but in water stress condition, it was more than 80. So just want to emphasize, and I think it's not really new. We had some data, so I wanted to show you, but I think we know that if you spray any herbicide in water stress condition, its efficacy is going to reduce. Okay. Just take home message. I think there are a few points I want to discuss. First, I think to me, Feathertop Rhodes grass is likely to become a year-round problem. And we use like only 10 populations, but I think in the future, we may include more populations just to see whether they have adapted to this environment. We are actually not in farming country, but a tillage operation could be used to vary weed seeds. We don't want, but I think in some cases it could be used like if it is a strategic operation. We need to target soil seed bank. I think now our all future management practices should target soil seed bank and prevent seed corruption. Recently, I think Lindsay Ward, a farmer, Paul, an extension guy, and myself, researcher, we three came together and we wrote a small article on awareness about Feathertop Rhodes grass. You could, you're welcome to go and read this one, an Australian Grain, September-October 19 issue. It talks about different methods. How can you prevent dispersal of this particular weed? Also, emphasize the importance of controlling these isolated weeds or in fallows. Feathertop could be targeted using harvest weed seed control, but limited opportunity for barnyard grass. And I'm not. I think we need to find other ways. How could you make this? How could we use other, like for example, crop competition, so that these weeds are susceptible to harvest weed seed control tactics. Also highlighted the importance of early crop weed control. And to me, this we are talking about always competitive crop. I may be wrong, but I think I see their role after 3 to 4 weeks of planting. Initially, we should have a weed-free field, and that could be with the use of pre-emergence or residual herbicide. And then later on, your weeds, your crop should take care of your weeds. Crop competition, I think it's not new. We started agriculture through this crop competition. When we started agriculture using broadcast method, that was actually the best crop competition. So it's not new, but now we need to find ways to use it in a practical way. We are talking always about, when I discuss about narrow rows and wide rows, I hear criticism that, oh, moisture is not enough. Is moisture really an issue? I can see, planting gear that, okay, we can't really have this planting machine which can plant that narrows. But to me, if we are using the same planting density, for example, we planted 7 plants per square meter of sorghum. It could be like, even in 1 meter as well as in 50 centimeter, we use the same planting density. So how then this moisture is a problem? Anyway, that's only my point. So to me, there's more uniform distribution of plants in narrow rows. Okay. Also, because as I mentioned, our crop residues remain on the surface. What I feel, when we have these narrow rows, when we harvest this crop, we have got more uniform distribution of crop residue. So it means that will help, that will reduce loss of moisture. That's my views. Another one, cover crops, I did not use. I did not mention anywhere, but it comes, when we are talking about plants in fallow conditions. So do we have any feature of cover crops in our environment, especially this when we don't have any rain? It's all totally dry. So I think we need to see that one. And with that one, I think I like to mention this last slide. I have request for growers, agronomists, researchers. We are looking for different, weed biology research ideas. So if you want us, or if you think that, oh, there is some gap there, please shoot an email or text me, about, and also which weed species. With that one, and we will be happy to, do that type of research. With that one, thank you very much. Paul.
Thanks, Sonny. Thank you so much for that. There was a lot of information there, everybody, and I'm not sure how many questions we got down the bottom there, Shannon. But look, I've got a heap of questions. I'm not sure how we're going for time, Shannon.
Yeah, no, shoot, Paul. Ask away.
Okay, Sonny. we've got to use these other weed control options, that's for sure. And that research you've just done there and presented so well with Barnyard and Feathertop, you know, everyone's got to realise that facing this increasing problem of herbicide resistance, we've got to use other tactics, and called it the Weed Smart Big Six tactics, which you mentioned with harvest weed seed control and other things. We need to be using them in conjunction with the knowledge that you've given us. So that crop competition, that early weed control, you know, that early weed control, we've been trying to practice that since the 80s when I was a young fella. And it's quite remarkable. Here it is in 2019, we're still talking about it, and we still haven't quite grasped that principle because, like you said, we're on the herbicide bandwagon. But the stuff you've given us has been quite superb to act on, because like the Weed Smart principle is, we're still trying to maintain our crop productivity in the threat of this increasing herbicide resistance. So the knowledge that you've given us with all those things there is pretty impressive, and I'm pretty sure, Shannon, that this is going to be available on the Weed Smart website, and that people can access it and look at all those numbers for the future. Is that correct?
Yes, it will. It will be up on the Weed Smart website later today.
Okay.
Yep.
I've got nothing else except to thank Sonny very much, Shannon, on behalf of everyone, and we'll learn plenty, and we need to adopt it and use some of his research and knowledge that we can use for better weed control in the future.
Great. Thank you, Paul, and thank you, Bhagirath.
Okay, thank you very much.
Thanks, Sonny. Thanks, everybody.
Yep,
Bye.
thanks.